So now we can begin our webcast. So welcome everyone. My name is Morten from Anybody Technology, and I will be your host for this webcast. And together with me here at Anybody Technology, I have my co-host Arne Kies. And with us from Germany, we have our presenter today, which is Kilian Wagner. Kilian is a research master student at Laboratory of Biomechanics in Regensburg. And also with us from Regensburg is Sebastian Dendorfer, Kilian's professor. So here first I'll just do a little introduction on the Anybody modeling system in general, and then Kilian will make his presentation, which is titled Investigation of Muscle Activation During Active Seating. And after the presentation I'll just do some final words, and then we will have a questions and answers session. But before we get started, let's just do a little housekeeping. For those of you who haven't participated in these webcasts before, on the right-hand side of the screen you will see a small control panel. You can collapse and expand this control panel by clicking the small red button on it. And inside the control panel you will also find a questions pane. So if you have any questions or think of any questions during the presentation, don't wait until the question and answer session to ask them. Just ask them right away, type them in the questions pane, and we will try to address them at the end of the presentation. We might not necessarily get around all the questions, but if we miss yours, I promise we will try to get back to you on email. So don't hesitate, just ask your questions. Also, I have to say that we are recording this webinar, so we will make it available afterwards. So all of you who are attending here, that you will get an email with the presentation and a link for the recording. Yeah, so let's get started. Who is Anybody? Let's talk about that first. Anybody is, for example, Anybody Technology. This is the company which we work for. We as a software company maintain the Anybody modeling system. We sell licenses for it, we provide training, support, and we do a lot of consulting work for different companies around the world. But Anybody is also a lot of research, and there is a number of research groups around the world who excel in using the Anybody modeling system. First and foremost, there is the research group here at Aalborg University. This is the research group where Anybody Technology originally originated. There's also a research group in the US led by Tony Protella, and then there is the research group in Regensburg led by Sebastian Dendorfer, and these are the guys you're going to hear from today, so you can look forward to that. Let's talk about then what the Anybody modeling system is, if you're not already familiar with it. The Anybody modeling system is a software for simulation of musculoskeletal systems. So besides the software system, which you see a small picture of here on the right side, Anybody is also a lot of models, which has been contributed by research groups around the world. This is what we call the Anybody Managed Model Repository. And using these tools, you can make a lot of different types of simulations, which include motion capture, ground reaction force prediction. You can combine imaging of your patient into patient-specific models, or you do man-machine simulations. So the Anybody modeling system is really a software tool that you can apply in many, many different areas. What we are going to hear about today is mainly the two, which is product design and optimization and ergonomic analysis, I guess. So without further, I will now give the word to Kilian. So Kilian, I will gonna unmute you. Are you there, Kilian?
Yes, I'm here.
That's good. So Kilian, I will make you the presenter now.
Okay, thank you very much for the nice introduction. As you already said, my name is Kilian Wagner. I work in the Laboratory of Biomechanics, which is part of the Regensburg Center of Biomedical Engineering, and this belongs to the OTH Regensburg and the University of Regensburg. And we are located in South Germany. The title of my master thesis is Investigation of Muscle Activation During Active Sitting. Before I will start my presentation, I would like to show you the contents of my work. First, I will give a brief introduction why active sitting is important and what was the aim of my work. Then, for the participants who are not familiar with Anybody, I will say a few words about the Anybody modeling system and the Anybody managed model repository. And next, I will present the experimental setup, show the implementation of the chair in Anybody, the model setup and the simulation setup. And after that, I will present the results, discuss them, and say a few words about future work. So why is this work important? This work is important as sitting is the most frequent working and leisure posture. But sitting in a wrong way leads to discomfort or health disorders, as for example, back or neck pain. The diagram here illustrates that back and neck pain are the leading causes of workless. And therefore, active seats are developed which might have a positive effect. Here you can see several examples for active seat devices. The person here takes place on an exercise ball, while the other two are sitting on ergonomic office chairs. Up to the present date, the development of seats is based mainly on subjective evaluation, on prototyping, and on experimental testing. And with the use of computer-aided engineering, for example, multi-body simulation, ergonomic questions become quantifiable. And this is what we did in our work. We investigate muscle activation during sitting on a specific ergonomic office chair by using multi-body simulation. So now to the Anybody modeling system. I will give only a brief introduction, as you already said something about that. As you already said, it is a software for musculoskeletal analysis, which is based on multi-body simulation. And the principle of inverse dynamics is used, meaning we put in observed or assumed motion or posture, and then we receive the computed internal forces. And there are several application fields. I will not repeat them now. The Anybody managed model repository is a library of models that contains body models and application models. An example for body models is, for example, a full human body model, or the mandible body also exists. And application models place the body model in the right context, namely an environment or an activity that is wished to be analyzed. The models are developed by research projects at academic institutions or by Anybody Technology in cooperation with academic institutions, and then those models are maintained by Anybody. Now to our experimental setup. We have 5 healthy male subjects, with all having approximately the same age, height and weight. And we have an ergonomic office chair, which you can see in the figure. The particularity of this chair are 4 pendulums. You can see them here. Those pendulums are attached to the seat pan on one side and on the pillar of the chair on the other side. So this means only horizontal movement is possible and tilting is not possible. The wheels of the chair are replaced with plastic parts, so the whole movement of the chair is provided by the pendulums. For the trials, the test subjects have to adopt an initial position. This can be seen in the figure. They seat upright, and I will describe later why this sitting position is important. The feet are aligned on the ground via positioning stripes, and the hands are crossed in front of the stomach to avoid a blocking of the markers on the lower extremity and on the chair plate. And then we have 2 active trials. The first one is a circle trial. There the test subjects conduct a circular movement with the hip leading to an equivalent horizontal displacement of the seat pan. And the second trial is a cross trial, meaning that we have alternating one directional movement forward and backwards, and after that to the lateral direction, meaning first to the left side and then to the right side. And to compare the results of the static trial or trials, we have also of the dynamic trial we have also made a static trial. In our work, we use 4 different measurement devices. The first one is our initial measurement units of deltas, or short IMUs. This is a method for motion tracking, and it provides information concerning the spatial orientation by offering angular position. An IMU consists of at least a gyroscope, which measures the angular velocity, an accelerometer, which measures the gravitational field, and a magnetometer, which provides information about the magnetic field. I will explain later why 3 measurements are important. This measurement device has a sampling frequency of 148 Hz. The second measurement device is motion capture. This is the gold standard for recording motion, and there some read reflective markers are attached on anatomical landmarks. And those markers are then captured by several 2D cameras which record the position, and from this information you can compute the 3D position. And for this measurement device we have a sampling frequency of 200 Hz. And for our boundary conditions we have a pressure indicating film on the seat gun and one on the backrest. On the backrest we inserted also an additional layer of foam. And we also have 4 3-dimensional force measure plates in the ground. For the generation of the specific chair model and the above lying pressure indicating film, we used a 3D scanner, namely the Xbox 360 Kinect, for obtaining the surface. And there we received an STL file, which was then treated in MeshLab and inserted an LS prepost to receive the X, Y, and Z position of the nodes. And those nodes were then inserted in MATLAB, and we computed a surface function of the order 5. You can see this formula, this function here. And with the help of this function, we computed an equidistant node cloud, where each node is a cell of the pressure indicating film. The figures on this slide incorporate this process, which I just described. And here you can see the backrest nodes where each node incorporates the pressure cell, and those are 1,440 pressure indicating pressure cells. Until now anybody was not involved, but this changes now, and for our work we use the standing model as a basis, where we deleted some of the drivers and we inserted the detailed neck model for implementing the rotation of the head around the 3 axes. The normal neck model of anybody just considers an extension of the head. So in this way the dynamic of the head can be added. By using the detailed neck model, this results in a simple muscle model. So next we scaled the human to the height and the weight of the test subjects by utilizing a scaling law based on external measurements. And therefore the test subjects had to adopt a position that can be seen here in this figure. Sorry. Okay. The human body model which we use contains a total of more than 1,000 muscles. Each of the trial consists of 4 steps in anybody and several MATLAB computations, and I will explain those steps in the following. But before I continue, I would like to describe the model setup in short. In step number 1, we determine the initial sitting position. Then in step number 2, the pressure indicating film is implemented, and from now on the head and the hip are IMU driven. The IMU is the red sphere on the forehead. Then step number 3, we compute initial ground reaction forces, and in step number 4 the muscle forces are computed. You can see a red coordinate system This is the reference system for the IMUs, as they do not refer to the global reference system. Now we'll give some detailed information to the simulation setup. We have 26 markers for the positioning of the human. And here you can see several blue and green markers. The green markers are the position on the human where the markers should be, and the blue markers are the measured markers, meaning the position where the markers really are. We have 4 markers on the ground and 8 markers on the chair, where 4 of them are on the seat pan and the other 4 are on the backrest. As some anatomic landmarks are hard to palpate or imprecise due to the overlying muscle layer, we use 4 different weight functions. The LASI and the RASI markers, those are the markers which are on the hip, are often lost during the test. So the hip is driven by using the IMUs. And for the IMUs, the exact initial position is used to bring up the relative rotation. Therefore, the hip flexion is adopted until the green markers overlaps the blue, meaning the measured markers. This process can be seen on the right figures. And when they match, we have the position how the subject really was sitting. And the reason why the subject in the figure here is sitting that upright is that this is the aforementioned initial position. As already announced, the measurement of an IMU consists of the measurement of acceleration, of the magnetic field, and of angular velocity. So why do we need 3 measurements? We need 3 measurements because the integrated angular velocity delivers angles that exhibits a drift that is evoked by, for example, noise or a temperature bias drift. This drift can be seen here, especially here. And I will explain later what you can see exactly in this figures. The drift is the reason why we compute 3 rotations out of the acceleration and magnetic field data via projected angles. And for the computation of the angles, we use the ATAN 2. 2 angles can be computed out of the first measurement device, and then the last angle is computed by using the second measurement device. And as the magnetometer is rather imprecise, 2 angles are computed by using the accelerometer. But those curves or those angles have a noisy trend, and so we use a Kalman filter for obtaining angles that have no drift and are not noisy. This is what you can see in the 3 figures here. The blue curve is the gyroscope data with the drift. Then the cyan curve are the computed angles, which are quite noisy. And then the red curve is the Kalman filtered curve, which is, as I already said, not noisy anymore and has no drift anymore. But the data still refers to a local housing coordinate system of the IMUs. So we generated a process to implement the data into any body. And it is important that you have 1 reference sensor on the ground. You can see this sensor. You can see the sensor here in the red circle. And 1 axis of the sensor is parallel to the coronal plane of the human, which is marked in green. To align all IMUs, we have a rotation matrix. This can be seen in the formula here. We have a rotation around 2 axes, so now all IMUs have the same G vector orientation. And then we have a second rotation matrix, this one here, that all IMUs have the same orientation as the reference sensor concerning the magnetic field vector. Only for understanding the formula, the hashtag S symbol here incorporates the number of the sensor on the human. In our case, the sensor on the head and on the hip. Then the initial here in the index, this is the Kalman filter data, and then we receive rotated data which can be imported into any body. And as IMU data refers to an initial orientation of the segments, the aforementioned process of adjusting the hip is very important. The hip and the head are now IMU driven, and all nodes of the pressure indicating film are included in step number 2. So now we conduct a full kinematic analysis, so we know the position of the undeformed pressure cells for each time step. As all bodies are modeled as rigid bodies, we do not know the deformation of the seat pan while the test subject is sitting there, but we can compute it in MATLAB, and this process is described now. According to the human who is placed by using markers, we know the penetration of the rigid chair surface. So now we can generate a linear relation between the highest penetration and the highest pressure. But the deformation is not in global coordinates, as the seat lies anyhow in space. So therefore we pick vertices on the seat pan. Those are marked as gray spheres in this figure here. And those vertices on the chair, we have those vertices on the chair to find a plane on the seat pan and the corresponding normal direction thereof. And now the deformation can be computed according to the linear relation, which I have, which I told you before, in direction of this normal direction. And then we have in the right figure here, we have the deformed surface and the new normal directions of the surface. The pressure matrix is in our work only a weight function for distributing the segment masses from the skull to the thighs. And what we do now, we define some nodes on the human body. Those nodes here. We have on this node we will insert the forces and the moments which we measured in the experiment. In the buttocks we have 12 nodes, and in the human back we have 7 nodes. Now in the first step, the distance between those nodes and the pressure cells are computed, and then the minimal distance defines the belonging of the human nodes to the pressure cells. This process can be seen on the right side of the slide. Due to dearness, the number of the nodes in the human buttocks was minimized from 12 to 6. And the color in this plot here indicates the belonging. And then we compute the forces by splitting up the scaled forces in an X, Y, and Z according to the normal direction, and the distance of the human node to the pressure node serves as a lever arm, and so we can compute the moments. In the next step, the ground reaction forces in the heels and one of the toes are computed with a frictional and a normal force component. This forces can be seen as the blue lines here. And therefore we had a toolbox of anybody where the forces are computed by generating a muscle between 2 segments. And in the final step, the inverse dynamics is computed for obtaining the muscle forces. The force input data is here marked as the green bars. And due to the scaled forces of the pressure indicating film, we do not know the residual forces due to movement, as they are not considered in the input file. But as we have a kinetic coupling of the pelvis to the global reference system, anybody computes those forces on his own. Now to the results. In the upper row, you can see the pelvis position in the projected plane, meaning that we look, that you look from the top of the subject down to the subject. And as the chair is always on the same position, we can see some subject-specific preferences of sitting. For example, subject number 1 prefer sitting on the right side and subject number 5 on the left side. And despite of static despite of the static sitting, small movements are also detected. In the dynamic trial, we have 2 subjects, namely subject 2 and 4, which only conduct smaller movement, while subject 3 has higher movements. This is this can be especially seen in the circle trial. And for the cross trial, we have a sideward deviation in forward and backward movement that is higher than the deviation for lateral movement. And we have higher movement to the right side than to the left side. In the box plot, the maximum muscle activation for each subject is shown. And one can see that we have almost constant maximum muscle activation in the static trial, as almost no motion occurs. And the maximum muscle activation is highest in case of the cross trial, where also the span between the upper and the lower whisker is higher. We investigated also the activation of a defined muscle group, and this muscle group consists of the erector spinae, the multifidi, the obliquus externus, the obliquus internus, the psoas major, the quadratus lumborum, the semispinalis, and the thoracic multifidi. This forms a total of 213 muscles. The exact location of those muscles can be seen on the following slide. The bars which you can see on the right side are computed in the following way. Each muscle group consists of several muscles, and only the maximum activity of one muscle is regarded. And then again, only the highest activation of all muscle groups is picked. So then we can compute an integral of those muscle activities and divide it by the integral of the maximum muscle activation. So this means if bar has a value of 1, then the considered muscle forms 100% of the maximum muscle activation. And what you can see in this diagram here is that the considered muscle groups form in almost every case the maximum muscle activation. Only subject 2 is an outlier. In this case, the muscles in the cervical spine and the rectus abdominis, this is a muscle in the abdominal region, have higher activation values than the considered muscle group. But the smallest bar has still a value of 84%. In the next step, we investigated the maximum muscle activation of a specific muscle group. And this formula can be seen, how it is computed can be seen here. In the figures, the x-axis shows the time, and you can see that the subject had a static seating position from second 2 to approximately second 5. And on the y-axis, the y-axis indicates the activity of the muscle. A value of 1 means that the muscle is fully activated and can therefore not deliver higher forces. The blue curves in these figures are the maximum muscle activation of a specific group, and the red curve is the maximum muscle activation of the whole simulation, and therefore it is the same in all plots of one trial. And when the blue curve is equivalent to the red one, this muscle forms the highest activity. And this curves show us that the movement leads to an oscillating muscle activation, where local minima are smaller than the maximum muscle activation at static sitting. And it is obvious that the multifidi muscles, those are located here, and the thoracic multifidi, the muscle group here, are small activated, while especially the erector spinae muscles, the obliquus internus, and the psoas major muscles are highly activated. And if you would normalize the blue curve by dividing it through the red one, you would receive a curve that is independent of the activation value. This is what you can see on the following slide. The formula can be seen on the lower right side. This figure contains much information, as it is for all subjects for one trial. So I will explain what you can see here. If a value of 1 on the y-axis is reached, this muscle forms the maximum muscle activation. So in this figure, the trend of a muscle group is very important. The nearer the activation value comes to 1, the higher is the part on the maximum muscle activation. So in this figure, in these figures, you can see again that the multifidi and the thoracic multifidi have minor activation for all subjects, while especially the erector spinae and the psoas major, those two muscle groups here, form the magnitude of the maximum muscle activation. We also took a look at the muscles on the right and the left side of the human body, and we found out that some of the muscle groups have always the same activation on the right and the left side. Here again, the multifidi and the thoracic multifidi are an example therefore. But we found that some of the muscle groups contain parts where muscles have a local maximum on the one side and a local minimum on the other side. An example therefore is marked in red in case of the psoas major. Considering the pressure distribution affirms the aforementioned thesis of subject-specific sitting of the preferences, and the pressure distribution shows that the maximum is always the maximum of the pressure is always on the opposite body half in comparison to the sitting posture. So now I would like to start to discuss the results. We found increasing maximum muscle activation from the static to the circle trial and then to the cross trial, and we suppose that this results from motion. And in case of the cross trial, it is highest if we have phases of acceleration and deceleration of the body mass. This is due to the non-continuous motion. And the smaller spawns between the upper and the lower whisker in the circle trial result most likely from the preservation of the motion. Considering the pelvis position showed that we have a higher deviation for a forward and backward movement, and the reason therefore might be that subjects do not brake after swinging back, but they use the swing to simplify the movement to the left side. And when they are on the left side, the movement to the right side is higher, as we have restoring forces of the pendulum, which sum up to the acceleration of the body mass. In the bar plot, we showed that the considered muscle groups forms a high part of the maximum muscle activation, and we suppose that there are 3 reasons thereof. We think that the considered muscles have a stabilizing function, they induce movement, and they have only smaller strength values in comparison to other muscle groups. So this means if we have a minor force value leads already to higher activation of the muscle. And according to our results, we conclude that the multifidi and the thoracic multifidi have only a stabilizing function, as they are only minor activated. And the higher activation in the other muscle group comes from their function to induce movement and or to maintain posture. And these results are in accordance with the corresponding function of the muscle groups listed in literature. So we can use those results for the validation of our model. Taking a look at the muscle activation on the right and the left side, let us conclude that the opposite-oriented muscle activation leads to one-sided movement, and there are 2 reasons for equal-oriented muscle activation. On the one hand, it may be a stabilizing function, and on the other hand, it may have the function for flexing the upper body. Concerning the pressure distribution, we found out that this results of a slight lateral bending of the hip, and the muscles then try to balance the translation of the center of gravity. And this is especially obvious in the psoas myoactivation. Now to potential benefits of this office chair. But here you have to mention that this experiment was conducted under laboratory conditions and that the duration of the trials are very short, with approximately 25 seconds. So this might influence the results. In literature, we found that an alternation of muscle activity possibly prevents the continuous activation of type 1 motor units. So this chair might prevent this continuous activation. And in literature, it is claimed that this might prevent back discomfort, which is experienced by during prolonged sitting. And furthermore, spinal shrinkage might be smaller in comparison to other active seating devices, as for example an exercise ball. As on one hand, we have a stable seating posture where tilting is not possible. And on the other hand, we have oscillating muscle activation that might have a positive effect on the diffusion in the intervertebral discs, meaning that we have also phases of smaller activation of the muscles than during static sitting, and this might reduce the diffusion. But this thesis cannot be verified as we do not consider an exercise ball, and we also did not take a look at the forces in the spine. Now to the conclusion, we found out that the considered muscle group from the values of the highest activation, as they maintain the posture or are used for conducting the movement. And furthermore, we think that sitting on an active chair might have some positive effects. And to conclude my presentation, I would like to say a few words about the future work. A long-time study has to be conducted to determine movement preferences on this active chair, and this could be done by an in-field study with IMUs, as they are not restricted to laboratory conditions. And what we already do now is we consider psychological effects, as for example stress on the muscles, on the muscle activation during sitting. So thank you very much for your attention. If you have any questions, you can write me an email. The email address was on the first slide. And if you would like to contact our laboratory, here are the contact information. Thank you very much.
Thank you very much, Killian. That was a really nice presentation. So I'm gonna take the presenter role back again. Yeah, that was a very nice presentation. Really impressive study with a lot of elements and some really complex models. So let's see, move on here. Before we get to the final thing about the questions and answers stuff, I think I'll just say a few words about our webcasts in general. If you want to look into more of these webcasts, you can find them on our YouTube channel. It has all the previous webcasts, and many of them are really, really interesting examples of using anybody and interesting research in general. We also have some upcoming events. For example, there is a free anybody workshop at the ESB in France, the 10th of July. You can find the registration link on our homepage. You can also meet us at the ICRA conference, the International Conference on Robotic and Automation in Stockholm, Sweden, here the 16-21 May. So if you're interested in that, you're welcome to send us an email. For any other events, publication lists, dates, all these kind of things, please visit our website. You can find all of these things there. And for users, there is also a forum site and a wiki site. This is the site that's called anyscript.org and has a lot of information for users. So this I think brings us to the end, the time for questions. So Killian, I'll just unmute you again. So we have we have some questions which have come in, but first there might also be some questions which I will just answer. For example, there is one that asks how this ground reaction force prediction that you're using for the feet, how is that done in more detail? And I think this is not a question for you really, and it's also a question which I cannot answer in detail here. But on our website, on this YouTube channel, you will find a previous webcast on ground reaction force prediction. It has a lot of detail on these kind of things. So I will suggest that you check this out. Then we have another question which you might be able to answer, Killian, which is along the same lines. This viewer asks, where you're not supposed to include the ground reaction force you measured directly into your Anybody model to get a proper muscle force, rather than the Anybody software to estimate those ground reaction forces. And I assume they mean the contact force between the feet. So maybe you can answer that, Killian.
Pardon, I didn't get the question.
No, but if you open your questions pane, you might be able to also read it. The question was where you're not supposed to include the ground reaction force you measured directly into Anybody to get a proper muscle force, rather than the using the Anybody software to estimate those ground reaction forces. But I guess you didn't really measure the ground reaction forces on the feet.
Yes, we did. We did measure the ground reaction forces on the feet.
Yeah. Okay.
But to implement we scaled the forces in the pressure indicating film on the seat pan and on the backrest, and therefore we also wanted to scale the forces in the ground, and this is why we computed an initial force for the ground.
Okay. Yeah, that makes sense. Another question we have here is how well do you think these results apply to seating in a car or a truck with steering wheel and pedals, for example?
It is hard to say anything about it without simulating it, but we have you have oscillating sitting position, so this might be similar to sitting in a car, for example.
Okay. Yeah. Do you have any idea?
You should simulate that for having a detailed for having a detailed look at that.
Yeah, of course. Do you have any idea why we don't see that many active seating products, for example, in car seats or in other products? Why that is not something that is included in all office chairs, for example?
I think it is hard to conduct to implement it into a car design, the active seating device, and the safety of the cars might be influenced.
Yeah. Okay. Makes sense. We have an asker here who's asking, did you use the IMUs to trace position of the limbs since the markers were lost?
Yeah, we did. We had the markers on the hip were we had problems with the markers on the hip, so we did not use them. We only used it for the initial position, but they were not implemented as drivers, but only graphically to put the direct orientation, to put the model in the direct orientation. So the IMU was the driver of the hip segment and the forehead.
Yeah, I'm sure many of the listeners might want to know how easy it is to integrate these IMU measurements together with marker-based data, because my experience says that this is not something that is out of the box, as you could say. How much work have you spent on doing these kind of things?
The implementation was quite easy. The difficult part was to obtain the input data which we can insert into Anybody.
Yeah.
But the implementation was no problem then.
Okay. The next question here is the direction of deformed seat surface considered when pressure distribution is attached to the model?
Yeah, it is considered.
How do you know how the seat deforms?
We know how the seat is deformed as we know the penetration of the human body in the hip with the seat pen, with the surface of the seat pen. The seat is more or less rigid, and the markers place the human body. So we have a penetration of the hip through the seat pen, and therefore we can compute the penetration, the new surface of the seat pen.
Yeah. Okay. That makes sense. I don't think there's any more questions coming in at the moment. So, so Killian, I think we will end the webcast for now. For all of you who are listening, we will share a recording of this afterwards, and you will also get PDF of Killian's slides, along with all the information you need, for example, to contact Killian or Sebastian. So, so Killian, thanks very much for a really nice presentation, and for all of you people who are listening in, I hope that you will be back another time to see some of our webcasts. With that, I'm just gonna say good day to you.